Okay. So today we're going to talk about chapter 4, which is the energy of life. So all cells will capture and use energy. In this example here, many of the sloth's cells are extracting energy from food and using its power to power his muscles. But what is energy? Energy is the ability to do work, the ability to move matter. And energy in this universe is neither created nor destroyed. It is just transferred from one form to another. So in this example, this sloth would eat, and then he would break down those nutrients, and then those nutrients would get broken down further to basic energy molecules, which then can be used by his muscles in order for movement. This happens very similar in your own body. Now, energy does come in different forms. We have kinetic energy, which is the energy of motion or movement, and potential energy, which is the energy that is stored and is available to do work. So if you look at this example here, if you have a compressed spring that has a high potential energy, if you actually relieve the tension, it will extend the spring, and that is essentially the energy is now released in the form of kinetic energy, and you can transfer the ball. So chemical bonds are a potential energy. A bond is between two molecules. Okay, so molecules like glucose and triglycerides will store energy within their bonds. To release the energy, the cell breaks the bonds. So whenever you store energy, you're forming bonds. Whenever you release energy, you would be breaking these bonds. If the cell cannot capture the energy being released, it will then be lost as heat. So energy is not created nor destroyed. It is actually only converted from one form to another. Energy changes form within this biological system, but energy is never created nor destroyed. And this is according to the first law of thermodynamics. So here we have an animal who is eating nutrients. Those nutrients were produced by taking sunlight, kinetic energy, and undergoing a process of photosynthesis to generate sugars and other molecules like oxygen. The animal then eats those sugars and will then undergo cellular respiration, where he breaks down those nutrients and will release chemical energy to be used for various cellular tasks. So cells are able to use chemical energy. Energy from the sun cannot be used directly by cells. Our cells are not able to undergo photosynthesis. However, plant cells are. It must be captured, stored, and converted before it is usable for cellular work. Energy transformations are oftentimes inefficient processes as well. Notice that heat energy is lost at each step. Heat energy is disordered, which means it cannot be used or converted back to a useful form of energy. Energy input is required for life on Earth. It takes a lot of energy to stay ordered. Cells constantly use energy to build their molecules and structures, to perform chemical reactions, and to carry out life's processes. Entropy is the measure of disorder. The randomness of the universe can be described as disorder or entropy. Since heat energy is constantly being lost to the universe and heat energy is disordered, the entropy of the universe is constantly increasing because we're constantly adding to it. This is called the second law of thermodynamics. In cells, metabolism includes all the chemical reactions that occur. Chemical reactions are the rearrangement of atoms. And this can occur in two ways. One, what we call anabolic reactions, which is building complex molecules out of simpler parts, like monomers. So here we have a bunch of monomers, we want to put them together. That's going to require the input of energy to form bonds, and we have newly synthesized larger molecules. Catabolic reactions, this is breaking complex molecules into simpler parts. This breaks apart chemical bonds. So here we have a larger molecule, we want to break it apart. And by breaking it apart, we're breaking a chemical bond, which then releases energy. Chemical reactions can require or release energy. And again, this is what we either refer to as a catabolic reaction or an anabolic reaction. Another term for these reactions is when we look at an endergonic reaction, this is going to form bonds and build molecules which will require energy input. Again, an energonic reaction would be an anabolic reaction. An exergonic reaction would be a catabolic reaction, which is the breaking of bonds to release energy stored within the bond. Now, some chemical reactions will actually just transfer electrons. Most energy transformations in organisms occur in what we refer to as oxidation reduction reactions. Electrons are transferred from a donor to an acceptor. Within an oxidation reaction, you have an electron donor molecule, which will release electrons. When you release electrons, you release energy. You then have a molecule which will pick up those released electrons, and that molecule is referred to as the oxidized molecule. If you have a molecule that is an electron acceptor, that means that that molecule will accept electrons or accept the input of energy. And the molecule that accepts electrons is referred to as a reduced molecule, which occurred by a reduction reaction. So again, oxidation is the loss of electrons from an atom or molecule, therefore releasing energy. The energy released by oxidized molecule is stored in the electrons. The reduction is the gain of electrons by an atom or molecule, which requires energy. The reduced molecule is the energy stored within the electrons. Oxidation reduction reactions occur simultaneously and often referred to as redox reactions. What this means is that if you end up releasing molecules of electrons by oxidation, something has to pick up that energy. Something has to pick up those released electrons. And that's going to occur through your reduction reaction. Whenever you oxidize one molecule, you're going to have to reduce another molecule. This is because of the fact that energy is not created nor destroyed, and it is only transferred from one form to the next because of the first law of thermodynamics. Energy that gets released, quote unquote, oxidized, has to be picked up by another molecule, quote unquote, reduced. In most cells, we have what's referred to as the electron transport chain. This is a series of membrane proteins that will participate in the sequential linked oxidation reduction reactions. Photosynthesis and cellular respiration both use electron transport chains to accomplish their end goals. As energy is released, the cells will store it and use it in other reactions. Now, what is the main energy currency for a cell? That would be ATP. ATP stands for adenosine triphosphate. It is a nucleotide that temporarily stores energy. It is the form of energy that cells can use. All cells rely on the potential energy stored within the ATP to power chemical reactions. The energy is being stored between the bond of two phosphate molecules of the ATP molecule, right here. This is a high energy bond. The overall structure of ATP is adenine, which is a nucleotide base. This is the same nucleotide base that you see as A for DNA and RNA, a ribose sugar molecule, and then three phosphate groups, adenosine triphosphate. ATP releases stored energy. Removing the endmost phosphate group by hydrolysis releases the potential energy stored in ATP. The cells will then use this energy to do work. So by the addition of water, you are able to break the bond between those two phosphates, releasing the phosphate, But when you break that bond, you've released a lot of energy. Cellular respiration is a series of chemical reactions that release energy from sugar or other molecules, producing ATP from ADP. So this is when you take nutrients, you are able to synthesize ATP molecules by breaking down those nutrients. ATP is often coupled with chemical reactions. The breakdown of ATP is coupled with endergonic reactions. The energy released from ATP breakdown is used to power the endergonic reactions. So what happens is ATP will energize the target molecule, making it more likely to bond with other molecules. So if you have here two glucose molecules, ATP kind of comes in, the glucose is activated by the phosphate that got grouped to that by the phosphate that got added to it. Now, that phosphate will then make it more desirable to then form a bond between these two molecules. And that's what will end up happening, forming a longer structure and releasing the phosphate again. ATP can also donate phosphate group that changes the shape of the target molecule. By donating the phosphate, you can physically change the shape of, say, a transporter protein. The shape change will then allow, in this example, ions to move across the membrane. Now, we're talking about metabolism, anabolic and catabolic reactions. Anabolic, again, is making larger molecules from smaller ones, requiring the input of energy. Catabolic is breaking down larger molecules to smaller ones, therefore releasing energy. The thing that mediates all of these chemical reactions are what we refer to as enzymes. Enzymes are among the most important biological molecules that are typically found inside of cells. Chemical reactions in cells must occur very quickly in order to sustain life. Without enzymes, many reactions would proceed far too slowly, and that would not be sufficient for sustaining life. So what is enzymes? What are enzymes? Enzymes are, first of all, a protein structure. They are made by the cells. Enzymes are catalysts. Catalysts are meant to speed up chemical reactions. In this case, an enzyme, because it is made up of a protein, is referred to as a biological catalyst. And its main job is to speed up the chemical reaction without being consumed by the reaction itself, or by actually becoming part of the product. The substrate is the molecule that binds to the enzyme's active site. Now, the active site is a very specific site for the binding of a substrate. It is like a lock and key. If the substrate is not a perfect fit, it cannot bind to that enzyme and will not be processed by that enzyme. Once the substrate molecule binds to the enzyme's active site, this is where the chemical reaction occurs. The substrate is where the enzyme will act on. Once the chemical reaction occurs, the product molecules are released. The enzyme retains its original form. Products are what the reaction produces. Enzymes ultimately will speed a variety of reactions. Note that all enzymes break a single substrate into not all enzymes break substrates into smaller products. Some enzymes actually combine two substrates to form one product. Okay, so this goes both ways. You can either break down, or such as for a catabolic reaction, or you can put together or form a larger molecule, such as an anabolic reaction. Enzymes speed up chemical reactions by lowering the activation energy. The activation energy is the energy that is required to start a reaction. Without the enzyme, activation energy is high. When the enzyme binds to the substrate, the activation energy is lowered. And this happens because the enzymes will actually sequester the substrates to bind. So again, when the enzyme binds to the substrate, the activation energy is lowered. Now, some enzymes will require what we call cofactors. Many enzymes have partners called cofactors that help catalyze the reactions. Cofactors will participate in the reaction to increase the enzymatic activity. A cofactor is going to be a non-protein component of the enzyme. And metal ions and vitamins are oftentimes very common cofactors. Now, cells control their biochemical reactions. Only some chemical reactions are occurring in a cell at a given time. Enzyme inhibition is one way to prevent unneeded reactions from taking place. There are many factors that can affect enzyme activity. Cells control the rate of biochemical reactions by limiting the activity of the enzymes. There are also inhibitors that can typically bind to the active site of enzymes, or inhibitors that can bind to other parts of the enzyme, creating a change in the shape of the protein, which ultimately would prevent the substrate from binding. If the inhibitor binds directly to the active site of the enzyme, it is referred to as a competitive inhibitor. If the inhibitor binds to an alternative site, referred to as an allosteric site, and ultimately changes the shape of the active site, the substrate can't bind, that is referred to as a non-competitive inhibitor. Non-competitive enzyme inhibitors change the shape of the active site. The cells can produce molecules that bind to an enzyme outside of the active site to alter the enzyme shape and prevent the substrate and enzyme from binding together. And finally, competitive enzymes inhibitors block access to the active site. They actually bind to the actual active site. So cells can also produce molecules that bind to an enzyme right at the active site, keeping the substrate and the enzyme from binding together. In many cases, the inhibitor is the product of the reaction that the enzyme catalyzes. Once there is enough product in the cell, it would be a waste of energy to produce more of that product. And what ends up happening is that will then inhibit the overall enzyme from creating new products. Cells will use what we refer to as a negative feedback mechanism to control biochemical reactions. In a process of negative feedback, what happens is the product of the reaction slows the production of a product. So essentially, the end result turns off the initial stimulus. There is another type of feedback mechanism referred to as positive feedback. In the opposite process called positive feedback, the product of the reaction actually ends up stimulating the overall process. So this is more of a snowball effect where you end up creating more and more and more product, because the more product you create, the more you produce. By the way, out of the two feedback mechanisms, negative feedback and positive feedback, cells use mostly 95% of the time negative feedback mechanisms. Enzymes can be affected by a few other factors. One of the most important factors is temperature. Most enzymes are active within a range of temperatures with an optimal temperature at which their activity is greatest. For the human body, that optimal temperature is 37 degrees Celsius or 98.6 degrees Fahrenheit. If you increase the temperature, what happens is that will cause the protein structure of the enzyme to denature, meaning unfold, and the enzyme becomes inactive. Enzymes also have the optimal salt concentrations and pH concentrations. If your enzyme is exposed to too low or too high of a pH, that can actually denature the protein and lead to unfolding, which can lead to inactivity. Now, a cell's interior is chemically different from its exterior. It maintains this difference by regulating transport of dissolved substances, which are referred to as solutes, across its membrane. Regulating what's inside of the cell is an example of homeostasis. The cell membrane, which is also referred to as the plasma membrane, is the barrier. Only certain substances can pass through the cell membrane. Solutes enter and exit the cells by different methods, depending on two factors. One is being a concentration gradient, and two, the chemical nature of the substance. What is its polarity? What is its charge? What is its overall size? A gradient describes the difference between neighboring regions. So if you have a tea bag, the tea is more concentrated near the tea bag than in the rest of the cup, forming a concentration gradient. Gradients are directional, and typically gradients will move down the concentration gradient, since it starts at high concentration and ends at low concentration. Eventually, what will happen with this tea bag is it will migrate from the tea bag, the most concentrated region, to the rest of the solvent, here the water, to eventually try to balance out or equal out. That process is moving down a concentration gradient. Now, concentration gradients dissipate without energy inputs. This is how it looks after time has passed. This concentration gradient is uniform throughout the cup where there is no longer a gradient. The dissipation occurs by random motion. The dissipated gradient is an example of increased entropy. Now, maintaining a gradient does require energy. Maintaining gradients requires energy since concentration gradients has a tendency to dissipate. Crossing a cell membrane can occur in several ways. By passive transport, which means it's by down a concentration gradient. These mechanisms would include such as simple diffusion, osmosis, and facilitated diffusion. Or things can move across a membrane by active concentration. Active transport is going to move against a concentration gradient. In vesicles by exocytosis or exocytosis, or through transporter proteins. So let's talk about simple diffusion. Simple diffusion does not require energy. It is a form of passive transport which occurs when the concentration gradients dissipate across the membrane. You're essentially moving across the membrane from higher to lower. Now, only small nonpolar molecules can cross membranes by simple diffusion. The size and chemical properties of small nonpolar molecules allow them to pass through the membrane's hydrophobic barrier. This is impossible for large polar molecules. Now, osmosis is a form of passive transport, which is simple diffusion of water across a selectively permeable membrane down its concentration gradient. This also does not require energy. Water moves across cell membranes by osmosis, which is driven by concentration gradients. So if you have blood cell in an isotonic solution, which is like something like saline, no movement occurs. Water in equals water out. If you place a cell in a hypotonic solution, meaning just plain old water, what would happen is you have very high concentration of solids inside the cell with very low concentration outside. And this will end up causing water to move inside the cell because it wants to try to balance it out. This will cause the red blood cell to swell and eventually cause the cell to explode. If you place a blood cell in a hypertonic solution, such as salt water, where you have more solids outside the cell than you do inside, now what happens is any water inside the cell tries to move out. And in this case, the cell will shrivel up. So again, to kind of recap, in an isotonic solution, water moves equally into and out of the cells, equal solute concentration inside and out of the cell. In a hypotonic solution, water rushes into the cells from the outside because you have a higher solute concentration inside the cell. And in a hypertonic solution, water rushes out of the cell. This is because you have a lower solute concentration inside the cell. Now, osmosis determines water content in plant cells. Plants usually keep the solid concentration inside their cells lower than the outside, so that water enters the cells. Hypertonic surroundings result in the loss of water, shrinking the large central vacuole. This can cause cells to lose their turgor pressure, causing the plant to wilt. Now, facilitated diffusion is a form of passive movement. However, passive transport for facilitated diffusion will occur when the membrane proteins transport substances across the membrane. So in this case, the molecule is either too big to pass through the membrane, or it is polar and can't pass through the membrane. And you need to use a transport protein in order to accomplish that. Again, because this is passive transport, you're not using any energy. So just to kind of expand on this a little bit, the hydrophobic tails of the phospholipids in the cell membrane will repel hydrophilic substances. Therefore, ions and polar molecules must pass through a protein channel in order to move across the membrane. Now, facilitated diffusion can also produce ATP. Hydrogen ions move across the membrane and down their concentration gradients through a transport protein. The protein uses the potential energy in the concentration gradient to produce ATP. When we talk about cellular respiration and we talk about how the electron transport chain operates, we'll get into this process in a little bit more detail. Now, active transport across the membrane will have to require a membrane protein, but because you're going against concentration gradients often, so in this case, we're going from where inside where it's very low concentration to out where it's very high concentration, this will require the input of ATP. I like to use the example of a sump pump in a basement. If you have water outside of a house, it's going to naturally want to flow to the lowest point, meaning it's going to want to go into the basement. However, we don't want water in our basement. So what we typically have in place is a sump pump, which will actively pump that water out from the basement back up out into the house. The sodium potassium pump is an example of active transport. When we get to talking about the cell membrane in a chapter coming up in a little bit, we will talk about how the cell maintains what we call resting membrane potential. It's going to maintain resting membrane potential by essentially pumping out sodiums and pumping in potassiums in order to create this charge differential across the membrane. Because you're pumping up against concentration gradients, this is a form of active transport, and the cell will take its utilized energy as appropriate. Endocytosis, this is a process in which you can transport out of this in and out of the cell, or in this case, endocytosis is in the cell, using vesicles. So what happens here is cells will form vesicles as they engulf fluids and large molecules to bring them into the cell. And that can be moved down there or against their concentration gradients. So with endocytosis, what happens is you bring it in via a vesicle. For exocytosis, here you have a vesicle, goes to the plasma membrane, it fuses with the plasma membrane, and the vesicle will then eventually dump out its contents. This is exocytosis. Endocytosis would be the opposite. Exocytosis and endocytosis both require energy, and they are forms of active transport. So investigating life, energy efficiency is something that is oftentimes looked at, and it's something we're going to explore over the next few chapters. Okay. All right.